Good morning.
Good morning.
You all right?
I think we'll wait as usual until 4 start. Erica, it looks so lovely in your background. I'm guessing it does not look like that in reality.
That's my background. No.
Yeah. Hello.
It's miserable, rainy, gray.
Yeah.
So we're at Dunya's computer because mine is blocked. Zoom blocked me.
Oh dear.
Is it just you two joining from Erasmus today? Or?
Max is also joining.
Yeah, but he didn't figure out he didn't need to put a Teams link on. So he will join soon.
Okay. And I'm a little confused now because Wendy, you're the host of these Zoom meetings, but it does say it's recording. Like it said it was recording as soon as I joined. So I don't know who's going to get the recording.
Maybe Matt set it on automatic.
Yeah,
that it
maybe.
always records.
Okay. That would be handy.
Because apparently I forgot my password, so it didn't allow me to log in because I don't have Zoom on my computer, so I always join online.
Right. Okay. I'm not sure if we're expecting many other people today because Matt said he might not make it.
Yeah.
Well, then it might be a little bit shorter meeting, but because it seems that you made the agenda, Clementine.
Yes, yeah.
And the minute taker is USCL, and the
I'll
chair
take the minutes.
is University of Sydney.
Yeah, so I'm guessing that'll be me. I don't think anyone else will be coming. Oh. All right, I think I'll just get started now because oh, okay, there is somebody else from USyd. Exciting. Hello.
Hi all.
All right, let's get started then. So I'll just go through it in order as usual. Max, do you want to tell us about LinkedIn and where you got to with that?
Yeah, definitely. So together with Dunya, I made a first pass to LinkedIn page. It's still pretty bare bones, but it exists and it's open for people to follow now. It doesn't have any posts yet because I think that's something we should maybe crowdsource a little bit. And I was not able to upload the link to the GitHub because I cannot edit the GitHub. At least it seems like. Or I just didn't understand it. It's also possible, but I looked at it with Dunya, but I did not manage to do that.
Can you comment on any of the GitHub repositories, or is it just the monthly Zoom meetings one that you can't edit?
I don't think I can. I'm not sure how I would edit this. But maybe it's an understanding thing. But I thought I just didn't have the rights to edit it because I don't see any edit button or anything like that.
Can you give comments on a repository or something? So if you go to the monthly Zoom meeting.
I'm at the monthly Zoom meeting, and what should I see where I could give a comment?
So if you go all the way down, you can. Can you add a comment?
I don't see anything about comments now.
Okay.
Okay. I think
I will take a look with you, Max, later, or Dunya will take a look to see what's wrong in your settings.
Yeah.
All right,
So
great.
Matt has given me and Wendy admin access. So if there is an access issue, one of us should be able to fix it now. Hopefully.
All right, amazing. So I'm just trying to get a regular link because I get the admin link now for the LinkedIn page, but I thought I would just drop it in the chat. Maybe I can just
that would be good.
use the admin link. You see if you get it, and then otherwise I try to figure out if I also can get there without the regular link.
Do you know how many people can be an admin on the LinkedIn?
So there are multiple admin levels. So you can have really the people who can also admin the admins. And then you can have a lot of people who have like the posting and commenting rights. And I think it's we can at least have multiple people have the real admin rights to also add and remove writers. So it might make sense to have just some of the more senior members of the different institutions have those rights, and maybe they can also delegate that right temporarily to some of their more junior co-workers. And then that's pretty much everyone also has the posting, replying rights like that.
Yeah, that sounds good. If everybody can have posting rights, and then maybe ideally one person from each institution with admin rights. Okay, cool. And then for yeah.
So I think right now it's very empty, and this is not a page that will gain a lot of traction yet. But I think we probably should first have a little try to have a lot of followers from like our group. So we should just have a little to get the follower count up. Then we should make an interesting post, and then people should repost that to their own account, so that the people, especially the people with the large networks within this field, so people like Matt, like Wendy, reach out, and then it gets through everyone in the mycetoma world. It will get on their feed, and then probably we get a small influx of people following, and then we will have to see in what way we can keep updating, keep pushing these LinkedIn things via the personal channels to other people. And I think I can also
maybe it's a good idea. Ma will have her PhD defense today on the and of course she did a lot of MycetOS work, so her whole thesis is on MycetOS. Maybe we can put a link there that her defense is today or was today, depends if we do it before or after. And we share that so that then people can see that a PhD thesis was defended today. And we can make a link to the thesis as well. And of course, we put it on GitHub. So we can link it to GitHub as well so that people can take a look there.
Yeah, that's a great idea.
Interesting.
I think I agree with what you say, Max, about we should maybe make sure the pages are properly populated before we start trying to share it. Matt had a couple, well, I suppose they're kind of, we all have the same kind of ideas about what to post about, which is PhD is a good idea. Papers that we've published, even if they're not the most recent, but from the past 6 to 12 months. I think Matt said if you can post things that you're doing with students, and instead of just posting a picture like, here's students doing chemistry, if you can post to the GitHub page or the lab archives or whatever it is that you're using to share your data, that would be better. And yeah, I mean, I might make a separate issue somewhere where we can brainstorm or just kind of collate things that we're going to post so that you know, if nobody's posted in a while, we can have a look and go, oh, there's that paper we didn't mention. Let's put that online so that, you know, the page hasn't just been sitting empty for a while.
Mm-hmm.
I'm not sure what the first post should be. Does anybody have suggestions?
Yeah, I think the first post might have to be a little introduction of what is MycetOS, what do we stand for, who is a member, anyone can join, something like that.
Yeah, that's a good idea.
And especially if we then really have like this, for this first post, a bit of a coordinated effort that we first populate it, then we make this post, and then we try to have like pretty much everyone, but at least the high profile members share this in their LinkedIn environment as a repost. I think that might be a nice kickoff for the channel.
Yeah, I think that's a nice idea to have some of the members, like maybe like a little like, you know, Wendy is, you know, working on this and Matt's working on this at UCL, and mention some of the different institutions and countries so that people can see the, you know, lovely collaborative nature and all that. Would anybody like to have a go at drafting that?
I think we can do that from our end as well.
Max and I can do that together, and we'll put it in a separate issue.
Okay, that would be perfect. And obviously, you know, let us know if you need any information or whatever from whoever. Okay.
Might
Just
even be
kind of
nice to
oh, yeah.
you could almost have a series, I guess, from sort of each institution and sort of how they're sort of contributing to the project. I think that would be sort of nice to see, or there's some institutions and how they're all sort of coming together with the various steps in that pipeline sort of thing.
Oh, so like different,
I think that would be
on
really
Monday
lovely.
we post this.
Yeah, this is Peter
Rasmus does
Rayner.
this.
This is his contribution to the project. You know, this is Shane. He's using first year students to build a small library to contribute. You know, just so we can sort of see all the different contributions and where they're going, sort of thing.
Yeah, I like that. That's a nice plan.
I like this idea.
On the same topic, I started making a who's who page for MycetOS, which is on the general repository, the one that's called General Start Here. I was basically just looking up people's like university profiles, and if there was something there that I could copy as like a, you know, like one line description, I was stealing that because I wasn't sure what else would be helpful to include. I think Matt recommended trying to put profile pictures in, which if everybody is happy for me to steal their university profile picture, then that's quite easy to do. But yeah, and it's fairly basic. If anybody has suggestions on what would be more useful to put on, I can add that. But at least for now, we have kind of a list of, you know, some of the contributors.
Yeah, I think for me it's okay if you want to use the picture on the webpage of our university. It's still at least somewhat up to date. You could also do it maybe a little bit more uniformal. For instance, I think everybody has an ORCID. Yeah, so that we put the ORCIDs there as well.
Yeah, I was thinking that might be a good idea. And yeah, I will add some ORCIDs then. If I can't find one or something for whatever reason, I'll post about it. Okay, cool. I think that's our main general business. Got anything else? Yeah, that's everything. Okay. Just to skip ahead for a moment, Series 5, do we have any updates on where that's at? Because I haven't heard where
Yeah, so we're still screening a few of the compounds to complete the final data set.
Okay.
And that's currently ongoing in the lab. So I expect that within a month or two, all the last data points are there and we can then finalize it. I know it's just the majority of the data is there, but if we publish, then it's maybe good to have those last data points in simply so that they will be published somewhere.
Yeah, of course. So that's still basically in, pending experimental work. Series 2, I know that there are a couple of compounds, the ones I think mostly sent by Peter from the Uni of York that you guys are still testing. I'm guessing that's a similar time frame. Okay. I have written a draft of the paper because obviously, it's not that hard to add the information that we get from 5 or 6 compounds. I've written it very much like I've put everything in that I think might be relevant, but depending on where we submit it, we probably will not need as much biological information or as much chemistry or whichever way around. Probably not as much intro in whatever paper. But I'm not sure where we want to submit it at the moment because obviously the journals that I tend to think of are more, medchem, chemistry related. I know that our last paper, like the phenaramoles paper that Hung and Dimitri worked on, that was RSC chemistries or RSC medchem, I think. Yeah, and I don't know if we have a goal in mind for the Series 5 paper and if that will affect where we want to try with the Series 2 one. Did we have any plans on that?
In the beginning, we were focused a little bit more towards a medical journal, and later on it became a more chemical journal because there was also structural activity relationships in it, which wasn't there in the beginning. So I guess chemically it's maybe more relevant than biologically.
Okay. And I think that's probably what we would say, yeah, about Series 2 as well. So I guess for now, are we happy to focus on, medchem journals and see what they think of us? And yeah.
Yeah.
Okay. For the people who have more experience with chemistry publishing than me, which is, I mean, basically all of you anyway, but what would your first thought be in what journal to go for?
Maybe I can be RSC medicinal chemistry or maybe one of the ACS.
Is it weird not weird, but
Mm.
does it hurt your chances to try and submit a fairly similar kind of piece of work to the same journal?
No idea. I shouldn't think so, right?
No,
I don't know.
biological journals, it doesn't matter because you
Okay.
usually
Yeah.
keep a little bit to your to the things you did before.
Yeah. I
Yeah.
think the
Okay.
good thing about the I think the good thing about the RSC journals is that they are open access, I believe.
Okay, yeah, that's a good point.
Yeah.
Cool.
Yeah. Otherwise, maybe the Elsevier journals as well, the bioorganic and medicinal chemistry.
All right.
This.
Yeah, I will talk to Peter and Alice as well and see what they think because we're supposed to be talking about this paper sometime soon. But it sounds like we're all on the same page. What was the last thing I had to say about this? Oh yes. In the past, I don't know if we've lost Wendy and Dunya there, but
No, we're still here.
you've just gone out of your, automatic
Yeah.
blurting. How have you shared your manuscripts in the past? Because I have I've got a Word document to share. Is it simplest for me to just post that on an issue and whoever is keen to can have a look at it? Or is it simpler to send it to one or, to specific people to proofread?
We did do both ways. myself, I find it easier just to send it by email, simply
Yeah,
because if you
that's
use Reference Manager, that stays intact. If you share it via a shared thing or a Dropbox, then
it
usually all the
gets, yeah.
references get messed up.
Okay, I think an email seems a bit easier to keep track of as well. And then we can obviously share the draft once it's a bit more, once people have had a chance to look at it, so that everyone can see what it is, etc. I will probably share those emails very soon then. And I use Zotero as a reference manager. I don't know if that will transfer across
Yeah,
as
I
well.
use EndNote, but what I usually do is the one who's in the lead, and others use different. Even if you use the same, sometimes it messes up. You can also just simply put a PubMed idea or a reference as a remark, so that the one who has the central library adds it later on.
Okay. Well, I guess we'll see how well the Zotero transfers across, and yeah, if not, we'll figure out how to fix it up. Does anybody else have anything to say on series 2?
One note on the Zotero and sharing. I don't know if you
Yep.
have the automatic updating of your library enabled or disabled, but sometimes if you like remove one reference, it removes the entire reference library. That might not be the easiest way to send it to everyone. So you can, so I would probably keep it on automatically updating once you send it, because otherwise, once one person edits something, then no one can see where one exactly references to.
Okay, that is good to know. Thanks. But if nobody has anything else to say about series 2 for now, let's move on to series 7. So, at last look, Junior had finished the high-low screening of those NDM compounds, so the ones from Dr. Melander, and the ones that the UCED first-year students made. And I've written on the agenda that the ChemDraw has not been updated. I did start updating it yesterday, so there's now 2 different ChemDraws on there, because there's one that I was trying to make tidy, and then there's just the old one there because I didn't finish tidying it up. But yeah, the main thing is that the methylating that oxygen at the 2 positions, so going from the OH to the OME, does seem to just kind of ruin the activity. So we will need to be making phenols in the future. Shane, did you want to tell us what you're thinking?
I haven't yet, but it sounds like very similar. Who was it? I think it was. I think Peter was doing the same thing, just coming up with a better synthesis of the acetylamides. I mean, the reported procedures obviously work, but they're not fantastic for students. I think the phosphorus trichloride, that's pretty nasty stuff to deal with students. There's some other ones like EDCL and that sort of stuff, but a lot of them can be very bad skin sensitizers, and so you can have an allergic reaction. So there's obviously like a big and find something that is going to be suitable for working with undergraduate students. So that's the plan this semester. Hopefully come up with something, come up with some sort of methodology by June sort of thing, and then June we can sort of start to decide, see where the project's at and see where what targets we might propose for the students in that next semester. So there'll be synthesis in August this year.
Okay, that sounds good. I wonder, because I can see that, I mean, Peter will be back shortly, but he's mentioned that they are going to be starting back in February. Would it be useful to because I believe his students are master's students. I might be wrong,
Mm.
but
Mm.
is there anything that, you could get him to do with his students and then see how that would translate, or is it a bit too difficult to match up the
No,
levels?
like I said, I think it pretty much parallels with what he's doing, so it might be
Okay.
a chance. I've got a lot on my plate at the start of semester, so it might be useful to see what Peter comes up with first. And then if they're struggling finding conditions and to expand those conditions, and I can contribute to that, looking at some of those other conditions, or maybe with reagents that he perhaps doesn't have on hand.
Yeah, okay. That sounds like it'll work out nicely with timing then. With the compounds, I guess we don't need to discuss it too much if Peter's not here anyway. Is there anything in particular that you think we should be making?
I haven't dived that deep into it yet. The ones that I chose with the methoxy, I guess the aniline linker are known, because the idea was to see what effect that methoxy group did have, whilst trying not to change too much the rest of the molecule. So although negative results, it's at least gives us some information that phenol is important.
Yeah, they're
So
nice matched ones.
yeah. So still negative results are results, so we can at least sort of move forward from that and go back to that sort of phenol group. We've sort of confirmed that that's important, and look at now expanding that other side chain sort of thing. So
I'm sorry, I believe there were also compounds ordered as well, because there was a couple
but
that
they haven't come in yet. They
Oh,
have been
okay.
ordered for a while now, but because we ordered them from some websites that we don't usually order from, like the Tokyo Chemical something, they haven't come in yet.
Which
so
chemicals
it'd be
were
interesting.
those?
Those were from Series 7. I
You know where
don't know.
where their
They're in
names
the
and
list.
structures are.
They are in, I did enter them manually into the main
In the
spreadsheet.
master list? Okay, that's cool.
So we might see, maybe if we get results for them by June or something like that, that might also help direct what sort of leads and targets we might go for that next semester.
Okay, because in terms of looking at trying to tidy up the chem drawer, I basically started making a big massive chart that it is trying to look at the differences on which way. What was I trying to do? Oh yeah, we have a lot of differences on each of the two rings, but mostly the ones we've made so far they always have chlorine in that meta position. So I basically took any compound that had a meta chlorine and put them into one chart, and any compound that does not have a meta chlorine is going to be somewhere else for the time being. So that's the logic behind how they're ordered on there, if you're having a look and trying to figure out what the hell I was thinking. But if anybody would like to suggest a different way to do it, obviously feel free as usual. But yeah, I was just wondering if we have an idea of what compounds we might make that might suggest, that might mean that a different style of organizing them is better. But I think, we'll just wait and see how it turns out for now. Also on Series 7, so I'll try and add those compounds that you say are coming in the post. I'll try and put them onto the charts at least so that we can see them as, hasn't been tested yet, but it's planned at some point. I think Matt had some comments about the page itself, which Anthony, you might also have some suggestions on this. I've lost.
Yeah, so I just kind of put a structure image up, but we kind of need to I guess there is a slight backstory to these compounds, because I had the paper, Wendy's group, when they were screening those, was it a kinase library, that IMID compound? That's what.
I thought it was from the OnCode, the FDA.
Yeah, OnCode. Yeah,
approved
OnCode.
Yeah.
It wasn't the kinase. It was OnCode. The IMID compound is what kicked the whole thing into going.
Yeah, and
Because
then niclosamide, was that that Bernhard just noticing a similar structure and suggesting that?
No,
Or
that came up, I believe, Wendy, I think that came up in one of the MMV boxes, right?
Yes, so niclosamide was also one of the hits in another paper, and
okay.
we then focused a little bit on that. But of course, we're not chemists, so we didn't notice that they were so similar. So
Okay, that's cool. I can have a look and try and put a simple thing together of at least putting those papers in and saying, you know, this one came from here, this one here. I think for
and I think
procedures,
Bernhard made some niclosamide
analogs,
derivatives as well.
and then you guys tested them, or
Yes,
Yeah,
they
there
were
was a
published.
paper. There was a paper on that. In the Series 7 repository, there's something called papers.md. It has all 3 papers that mention salicylanalides.
okay, great. Because yeah, is it the superior anti-cancer and antifungal activities of new sulfonyl
Yes.
substituted nicosamide derivatives? Beautiful. Okay, I do have that one. Cool. But yeah, I can, you know, write some words around those papers and link them. Procedures, Shane, is that something you can put together just as essentially just like a, you know, here's what we actually did, moving what you probably have in another issue into the wiki?
Yeah, I think I did upload the ones that we did last year. So they're
Okay.
up in the wiki that the students completed.
All right, I'm guessing
yeah,
Matt
see
just
if we
didn't
come
look
up with
too
anything new.
closely. Yes, okay.
And then there's 2 procedures on the bottom I added that I found from literature about EDCL.
All right, I might maybe just add like a paragraph, or maybe even a table or something, Shane, because obviously some of these worked better than others.
Mhm.
Is that already online somewhere? Which one you kind of, you know, went with as the final one?
No, I didn't report the success of each of those. So I can either write up something about it or
Yeah, if you have time to just kind of, you know, write like I said, either a short paragraph or just a table saying good yield, not pure, whatever the reasons are that you decided to well, I suppose safety is also a big aspect there. So that if somebody looks at that page, they're not going, well, why didn't they just do the oxalyl chloride one? Why did they bother with these other 4? You know, or
yeah, sure.
whatever
Yeah,
they
definitely.
might think.
I mean, that was a conversation that we had in class, so we can sort of summarize those points and bring it in. And it was a
Yeah.
combination of safety, purity, and we also talk about green chemistry as well. So,
Oh, yeah, perfect.
those points.
Okay, great. And what else did Matt suggest that we needed to do? I think he had a suggestion about an ambassador for Series 7. SAR link to master list. I can put that in. Yeah, what do we think about the idea of having a Series 7 ambassador? I'm assuming from what Matt has said that he is thinking that this would be a new person, somebody that we, you know, convince to get involved. I don't know if that's how you guys interpreted it.
Yeah, so we of course have PhD students here, but I think this one needs a chemical lead as well. So somebody who understands the chemistry and the correlations between the compounds, and that's the part we
Yes.
cannot do.
I don't quite know the current state of honors students that Peter and Alice might have, but if they had a student who was going to work on open source drug discovery, even if they were focusing on a different target, I would suggest that we could ask them, because it's a kind of, you know, low impact way to get involved. I don't know how likely it is that we're going to just convince a stranger to you know, to do this kind of thing.
Yeah, because I don't think at UCL there's currently a starting PhD student on this subject, and also not in your university, is it?
I'm
because
not
this would
sure.
give a nice
Yeah.
kickoff.
Yeah, I'm not sure actually about UCL. I did ask Matt if any of the MSc students would be working on this project, so we could ask them, but I'm not sure. I'll double check.
And the other thing, which could be more trouble to organize than it's worth, is if you've got students working on this, then it could be something where you ask them to, write introductions essentially, and then we can choose one of those to be what we put on.
Featured.
Obviously, that's a bit, it kind of works once, doesn't necessarily work as an ongoing thing because your students are only around for a year.
Yeah.
But it is a nice way to, show the students that what they're doing is part of a bigger thing, which I think is a bit hard to get across sometimes.
Yeah. Yeah, so I'll check with Matt and see if we have an MSc, which they start very soon. An
They
MSc
start in
student, so
February?
hopefully. This year they start soon. I mean, but they are in March, but they are doing more of
Okay.
introductory things and stuff. The actual very focused lab work is mostly in June, July. Yeah.
Okay. I was also talking, so last year we did a I don't know, outreach thing with in Australia when you're in your final year of high school, you have to do these, well, some science students have to do these like in-depth independent chemistry projects, and we were working with a school and one of their students, and they were making an aminothiazole, which was obviously lab-based, and they were doing that at their school. That school's keen to keep working with MycetOS to do stuff, but I can't really, I'm in England now, so I can't really help in terms of physically, being there. And like I said, I'm not really sure if any new students will be working on mycetoma. So I did suggest to that teacher that we could focus on, something hopefully a bit more exciting than a literature review, but, being involved in the kind of background and understanding side. And then we can ask them about doing some of these things as well. But yeah, for now it sounds like keep your eyes open for people who
Which school was that, Clementine?
Barker College.
Okay. So I got
Yeah,
approached by
they-
another tech school. They've actually been looking for a medchem project to teach
Which-
HPLC.
Which school's that,
Uh,
if you remember?
yeah, I'm just quickly pulling up the details. Yarra
I
Rangers,
was actually, sorry.
Yarra Rangers Tech School.
Oh, I've never heard of them.
No,
I also-
neither did I.
Where's the Yarra Rangers? Surely that's
No idea.
Melbourne. Yeah, the Yarra
Um,
Rangers themselves are in Melbourne, but Helston Park Agricultural School is not in Helston
Box
Park.
Hill?
So,
Box Hill.
Okay.
So yeah, so they've got HPLCs that they use. They teach looking at caffeine and parabens and that sort of stuff, and so they want to develop a medchem program to use their HPLC. So I was possibly thinking of sending the samples that we make. We do HPLC analysis on it for
Yeah, that
purity
would be
and log D determination. So maybe they can be part of that part, and we can just send them some sample to analyze and contribute that way.
Yeah, I think that would be a great idea. And I mean, you could also send them the intermediates and things if that gives them a bit more stuff to work on.
Yeah. So the person I've been speaking to is on holidays until I think about sort of February sometime. So I'll catch up with them and sort of see what the possibility of that is to have them on board.
Yeah,
That's good
okay,
because a lot of the agricultural chemicals, like the SDHI type classes, are very similar in structure. The SDHI
So-
type herbicides, a lot of them are diarylamines, very similar.
Yeah. Sorry, Peter, you missed us mostly talking about Series 7, where we were saying that Shane is planning to make compounds in semester 2, which starts in July or August. But we can see that you're making things sooner than that. So if you have any specific thoughts already on what you're going to make, let us know. But apart from that, we were discussing the admin around Series 7.
Yeah. So we tried making some straight coupling with the just straight from the salicylic acids just before Christmas, and they don't really work very well. So what we were planning to do is we've got maybe 10 lab days left with these students. So we were planning to do some method development to try and get some robust methods. I'll post the structures that we're using to optimize those methods, but we weren't planning on making loads of them.
Yeah. Are they ones that are novel that we haven't tested yet, or a mixture? So
there are one that is currently one of the hit compounds, and then a few derivatives of that. So I'll post them in the group, but there's only probably 3 or 4 of them that we may or may not end up making, but I'll keep you updated. We're hoping just to get a decent method. But yeah, that's our current plan on Series 7.
Great. And that sounds like it works out well. If you guys do figure out a nice method, then that'll work out nicely with our UCID timeline.
Yeah. Great.
When do you think you'll have the results of them done, Peter?
Certainly so the students have to submit their final reports in April. So, but I'd imagine we'll know in the next month. Things are progressing.
Yeah.
So yeah.
No worries. Yeah, just let us keep us up to date, and then if none of those work, then I can start working on extending other methods to see what might work.
Yeah, that sounds great.
All right, great. And we're at the end of our agenda. Does anybody have any other business? Oh, is there? What have I forgotten?
So Tim left, obviously, so he's not here anymore, but there's a replacement.
Oh,
wait. Saskia is here as well.
Hi.
Hello.
This is Saskia. She's taking care of what Tim was supposed to do, and she is my co-promoter, and she will join the monthly meetings now as well.
Okay, great.
Hi, nice to meet you all.
Okay, do you already have a GitHub, username?
Sorry?
Have you got a GitHub login
You're
yet?
already in GitHub.
I was, because I was in Wendy's lab a couple of years ago, but I'll have to look it up. It's probably not active anymore, I can imagine.
Okay,
So I will
just let us know.
we'll see. And otherwise get registered again.
Okay, great. And so you'll be working on the what's the word?
Knockouts.
That's knockouts. Okay,
Yeah,
cool.
exactly. Your molecular biology work.
And she also is the one who was helping to make we had some target proteins selected last year.
Yeah, the kinases.
Yes, at UCL, they are making them from Aspergillus, and we are making them from Madurella, so that they can later on be screened in the Structural Genomics Consortium, so that we could identify some potential leads specifically for the proteins we identified. So it's one kinase and it's one SIDRA4 gene.
Yes, I did see actually, Matt said samples of SID-A and MKK2 are still being generated. And does not have a more specific update than that for now. Okay, great. Okay, so just to quickly recap, we have a beautiful LinkedIn page now, which I can post on here if Max still, if GitHub is still being difficult. Everyone can join as a posting with posting rights, but we'll try and have at least one person from each institution. I'll update those Who's Who pages with some ORCID profiles as well as pictures, unless anybody objects. And I should have said as well, if you have a picture that you'd rather that I put up instead of whatever's on your, institution page, just either add it yourself or just let me know and I'll put that one in. I know some of them are, it's just hard to update them. through uni. And I don't have one anyway, so. I'll share the draft of the Series 2 paper, I guess probably with Matt and Wendy for now, unless anybody else would like to see it, you know, in the first pass. I'm just waiting for Peter and Alice to have a look at it. Series 5, we're still testing things. Series 7, I think we probably all remember that conversation still. And yeah, the other major point was posts that we can make for LinkedIn. I'll make a separate issue so that we can put some ideas and things there. And if you have stuff to pop on, put it there. I did, especially with those ideas that you had, like Shane and Max, about having a who's who. What do you think about having some kind of template? Like, I was thinking like a Canva template, except I personally don't like Canva.
So you mean like really like a format, like a certain background, and then with the picture here, and then named double points, something like that, or?
Yeah, that's all I was thinking. And then especially if you say, like, if we do like a, you know, who these people are, just so that it's, you know, if you guys make one at Erasmus and you do it in beautiful pastel pink and blue, and then we have one from UCIT that's green, it just maybe looks a bit odd. I can have a go at that, but also if we think it's too much effort, I'm happy to let it drop.
It might be easier to have at least a form that, you know, we fill out and we just sort of fill out a quick sort of bio.
That's a good idea, yeah.
And about sort of projects. And then it could be just something that you could just, even just AI generated into Canva or something other sort of program.
Yeah, I mean,
even like a PowerPoint page.
Yeah,
Yes.
Yeah, that's right. So at least you can sort of mostly copy and paste and just sort of reformat.
I like to be old-fashioned and use PowerPoint because I find it just kind of, you know, when you try and edit something, it normally does what you want it to. And Canva does not do that for me. But so, I mean, obviously, you know, name and affiliation, institution, whatever. What else would we like to include on a who's
maybe
who?
Just a short sentence on what you're working on.
Yeah. Like relevant to mycetoma and more generally, like one that's what you do generally, and then one that's the specifics, perhaps.
Yeah, sounds good.
Yeah.
I
Anything
think we should
else?
be open as well, because I mean, you've got, I think have it open for other things like schools, maybe even if there's general public contributors who are, you know, actually participating in citizen science. You know, it needs to be inclusive enough that, you know, that it's not institutionalized, that it is, I'm a general citizen from Norway
That's
and
a good
I'm
point.
interested in this and I'm contributing by, you know, reading articles and providing the latest data or trends or suggesting sort of stuff. So I think to be able to sort of accommodate those will ensure the citizen science part of it.
And then we could get students, especially, maybe 65 first years is a lot.
Yeah, that's right. Well, yeah, well, we could put a call out, you know, that we want a student representative to post their
that would be
view
great
on the project sort of thing. I think when that comes around, I think it'd be great. Peter might do the same with some of his students as well.
So something maybe like name and like location, but then we can have affiliation and I mean, for want of a better word, like title as optional things. So, you know, you can say high school student, but you also don't have to put something in there if you know it's not relevant. How they're helping maybe, or how they're
contributing to the project. Yeah.
And what they do. Okay. Yeah, that sounds like a great plan.
Right. Something slightly different that I also looked into also on the more technical presence side was the Gmail account we have now can only be entered by one person. And I looked into if we can maybe have like an inbox that can be accessed by members from all kinds of institutions. And I searched myself and with the IT department of the Erasmus and with the IT department of our department. But the answer is no, we cannot do that. So that's just, as long as everyone is enrolled within the same institution, those shared inbox things do exist, but it's not something that we can actually arrange in a good way. Especially now that everywhere you have the multiple step verifications requirements and everything. I think the added benefit of having like one location have a shared inbox, but still the others cannot access is too minimal to try to set that up over the system we have now. So I think that was just not feasible.
All right, thank you for looking into that. But RIP.
Yeah.
Yeah. I think we will still edit when Matt is at a meeting. We should still definitely talk about who has access to that account then, because I think Matt probably is not the best place to sort through every email. But yeah.
Yeah, and maybe we should also just have a point on the agenda that the person who is in control of the inbox discusses the mail that we got. And if there's nothing that's just very briefly skipped over, or if it's just, but if there's interesting stuff, that at least there is a reminder every month that someone should maybe check the inbox.
Yeah, that's a great plan. I like that. If that's the last point, then that might nicely segue to who is the chair for next meeting, because we can add it in as the first thing for that agenda. I'm assuming it's either UCL or Erasmus, but
I did make a new version of the file. It's in the bottom, Dunja. Yeah, it's the ene
the
laatste.
20. There we go.
Lisa.
Yeah.
Okay, so UCL is chair. So, well, Max, you can put it on the agenda yourself then, if we get GitHub to work.
UCL, you say? I'm not UCL. I'm
Oh,
Erasmus.
sorry, you're right. Not thinking. All right,
That's
then.
okay. No worries.
We will add it.
Thanks,
Great.
Erica. Can't complain that it's the late hour now.
It's too early.
Okay, so I guess we'll see each other on the 24th of February with UCL as chair next time. And Erica, I'll put the recording up, obviously, and Erica, if you pop the minutes up whenever you've got them ready.
Yeah.
Yep, cool.
Yeah.
And I posted the links on the PhD defense of Ma. It's a hybrid, so it's also online. So in case
so
anyone would like to join, they're underneath the agenda and at the point where we all put the thesis in. So I also put a copy of our thesis in.
Okay, great. And so when you say you put it on the thesis, is that on I think it's the general page?
It's the page where we have it's the talks, meetings, publicity. There's also a feature called PhD thesis.
Okay, I will have a look because I think maybe we have some cross-posted theses, which is not really a big issue, but I'll just make sure that...
No, I saw that Wilson's was there, but that was the only one. So maybe there's also another place where we put thesis.
Yeah, I put Hung and Dimitri's on at some point, but I didn't know where Wilson's was. So I'll have a look and make one of them the main one.
Okay.
But as long as they're all there, that's fine. Great. Okay. Well then, see everybody next month.
Okay.
Bye.
Bye.
See you all later.
Bye. See you next month.
Bye, everybody.